Chapter 2: Servers and Linux, the foundation of bioinformatics computing. In the previous chapter, we introduced bioinformatics as the intersection of biology, computer science, and statistics. But to actually do bioinformatics work, you need the right computing infrastructure. That starts with understanding servers. So what exactly is a server? A server is a specialized computer designed to provide services to other machines over a network. Unlike your personal laptop or desktop, a server is built for heavy workloads and reliability. Think of it as a workhorse that runs around the clock, handling requests from many users simultaneously. Servers have several key characteristics that set them apart. First, they have high-speed CPUs optimized for intensive computation. Second, they are designed for long-term reliable operation, running 24 hours a day, 7 days a week, without interruption. Third, they offer strong input-output throughput, meaning they can read and write large amounts of data quickly. And fourth, they provide expandability, allowing you to add more memory, storage, or processing power as your needs grow. Now that we know what servers are, let's talk about why bioinformatics specifically requires them. There are three main reasons. The first is compute resources. Bioinformatics datasets are enormous. A typical laptop has 8 to 16 gigabytes of RAM, but assembling even a relatively simple fungal genome can require 100 gigabytes of memory. This is not just a matter of speed. If your machine does not have enough memory, the analysis will simply fail. It cannot complete at all. There is no workaround. The second reason is disk durability. Bioinformatics workflows involve reading and writing massive files repeatedly. This puts enormous strain on storage drives. Servers use RAID arrays, which stands for Redundant Array of Independent Disks. RAID spreads data across multiple drives, improving both read and write speed and providing fault tolerance. If one disk fails, your data is still safe and recoverable. The third reason is continuous operation. Some bioinformatics analyses take a very long time. A large genome assembly might run for days or even weeks. Servers are designed to run uninterrupted for these extended periods. A laptop, on the other hand, might overheat, run out of battery, or need to be restarted for updates. You simply cannot rely on a personal computer for multi-day computations. So how much computing power do you actually need? Let's walk through a real-world example. Consider a human whole genome sequencing analysis at 30x coverage using Illumina technology. This is one of the most common bioinformatics workflows. First, the raw sequencing data. Your paired-end FASTQ files, even when compressed, take up about 86 gigabytes. After quality filtering and cleaning, you still have around 48 gigabytes of clean data. Then you need the reference genome and its indices, which add about 5 gigabytes. The alignment step is where storage really explodes. Going from raw reads to a final analysis-ready BAM file involves several steps: alignment to SAM format, sorting, marking duplicates, and base quality score recalibration. If you keep all intermediate files, this stage alone consumes about 372 gigabytes. Variant calling adds another 8 gigabytes for VCF files and intermediates. In total, if you keep only the essential files, you need at minimum 193 gigabytes. But if you keep all intermediate files for quality checking and troubleshooting, the total reaches 532 gigabytes. And that is just for Illumina data. For Nanopore sequencing, the situation is even more extreme. Raw FAST5 signal files are approximately 10 times larger than FASTQ files. A 45x human genome sequenced on a Nanopore platform requires about 3 terabytes just for the raw data alone. Let's compare servers and personal computers side by side to make the differences concrete. In terms of use case, servers are designed for shared services and continuous operation, while personal computers are meant for individual use. For CPUs, servers often have multiple processor sockets, giving them far more computing cores than a personal computer's single CPU. Memory is another major difference. Servers can hold hundreds of gigabytes of RAM, sometimes even terabytes, while a typical PC has 8 to 64 gigabytes. For storage, servers use RAID arrays with high-speed input-output, while personal computers typically have a single solid-state drive or hard disk. Servers offer hot-swappable components, meaning you can replace parts without shutting down the machine. Personal computers have limited expandability. Servers support many concurrent users working simultaneously, while a PC is designed for a single user. And for reliability, servers include ECC memory that detects and corrects errors, along with redundant power supplies and cooling fans. Personal computers use standard components without these safeguards. The trade-off, of course, is cost. Servers are significantly more expensive than personal computers. Now let's shift our focus to the operating system that runs on these servers, Linux. What is Linux? Linux is an operating system created by Linus Torvalds and first released on October 5, 1991. The name is a combination of Linus and Unix, the earlier operating system that inspired its design. You might see a cute penguin mascot called Tux, which is the official symbol of Linux. Linux has several key advantages that make it the dominant operating system for scientific computing. It is open source and free, meaning anyone can download, install, and modify it without paying licensing fees. This also means it can run on an enormous variety of hardware. It is remarkably stable and efficient, able to run for months or years without needing a restart. It supports multi-user and multitasking environments, which is essential when many researchers share a single server. and it provides high security through its permission system and active community of developers who quickly patch vulnerabilities. For bioinformatics, we almost exclusively use the command line interface rather than a graphical desktop. This might seem daunting at first, but the command line is actually more powerful and efficient for processing large datasets. Linux comes in many different flavors called distributions. A distribution combines the core Linux kernel with bundled software like package managers, desktop environments, and utilities. Let's look at the most common ones. The Debian family includes Ubuntu and Debian itself. These distributions use the apt package manager, which makes installing software straightforward. The Red Hat family includes CentOS, Red Hat Enterprise Linux, and Fedora. These use either Yum or DNF as their package manager. There is also OpenSUSE, which uses the Zyper package manager. For bioinformatics, the most relevant question is usually Ubuntu versus CentOS. Ubuntu tends to ship with newer versions of system libraries like GCC, glibc, and zlib, which can make compiling and installing some bioinformatics software easier. However, if you use Bioconda, which is the most popular package manager for bioinformatics tools, the difference between distributions becomes negligible. Bioconda handles all the dependencies for you, regardless of which Linux distribution you are running. So why should you learn Linux for bioinformatics? There are 5 compelling reasons. First, most bioinformatics software only has a Linux command line version. Tools like BWA, SAMtools, GayTK, and countless others are developed for Linux first, and many have no Windows or Mac versions at all. Second, Linux is open source and free, which aligns with the open science movement in research. Third, high-performance computing clusters, which are essential for large-scale analyses, all run Linux. Fourth, Linux supports multi-user and multitasking environments, so many researchers can share the same server efficiently. And fifth, the command line enables powerful automation and batch processing. You can write scripts that process hundreds of samples overnight without any manual intervention. However, I should be honest, Linux can be challenging for beginners, and there are 4 common pain points. The first is the command line interface itself. There is no graphical point and click. Everything is done by typing commands. The second challenge is understanding the directory structure. The single most common error in bioinformatics is getting file paths wrong, resulting in the dreaded no such file or directory message. The third challenge is environment configuration. Different Linux distributions and even different servers can have inconsistent setups, which leads to software installation headaches. And the fourth challenge is permission management. Multi-user servers require strict access controls, and understanding file permissions takes practice. Don't let these challenges discourage you. With consistent practice, Linux becomes second nature. That wraps up our introduction to servers and Linux. You now understand why bioinformatics requires server-level computing resources, how servers differ from personal computers, and why Linux is the operating system of choice for this field. In the next chapter, we will learn how to actually connect to a remote server using SSH and start working on the command line. If you found this video helpful, please like and subscribe for more bioinformatics tutorials.